All right, thanks very much, and it's an honor to be here, and it's to meet with the families, it's to meet with the scientists, to meet with the advocates, and an honor to speak right after Kozo, who started the molecular biology of this field. So I'm Andrea, and a couple things about myself. I don't have a slide. So I'm a professor at Harvard Medical School. I did a pediatric training at the Children's Hospital of Philadelphia, CHOP. We have a gentleman here from CHOP. And then I went on to back to Boston to Harvard, where I have been working for 30 years. I'm an oncologist. I focus mostly on blood cancers, and but I've become over the years an expert in the area of DNA repair. I actually gave up my clinical profession about a decade ago. And what I found is through this systematic study of DNA repair, I've identified a number of genetic syndromes which have defects in DNA repair, and I think that the CHAMP1 syndrome is one such feature. I've been working for 30 years actually for the Fanconi Anemia Research Society. Actually was recently given the Lifetime Achievement Award for working with that syndrome, and recently been elected to the National Academy of Sciences from the basis of that work. But I'm honored now to work on CHAMP. So I'm going to tell you about CHAMP1 and Fanconi anemia and what I believe to be two related genetic syndromes. So what I'm going to tell you is some clinical features of this disease, Fanconi anemia, which is a DNA repair disorder. A critical protein in this Fanconi pathway called Rev7, which Kozo just referred to. Tell you about how CHAMP1 binds to this Rev7 protein, really demonstrating that there's crosstalk between these two syndromes. And finally, the CHAMP1 syndrome is indeed a DNA repair disorder with distinct features, as you well know, developmental defects, intellectual disability, and with other clinical consequences, and that's what I want to talk to the families about. All right, so Fanconi anemia, something that you don't know very much about. It's also a rare disease, though considerably more common than CHAMP1 syndrome. It's autosomal recessive. In this case, the parents are both carriers of Fanconi anemia mutation. So there's a 1 in 4 chance that a child in the family will develop the disease. These children have developmental defects. They have some facial abnormalities. They do have a bone marrow or a blood disorder, which I'll tell you about, and they actually develop cancer. And the cells from these children with Fanconi anemia are very sensitive to DNA damaging agents. Actually, Kozo just showed you that cells with a defect in CHAMP1 are sensitive to radiation, and that's true for Fanconi anemia as well. That's one common feature. So these children have a very characteristic physical appearance. They have some funny spots on their skin we call cafe au lait spots. They have some skeletal defects of their thumbs, and they develop various kinds of blood syndromes, and that's why I'm particularly interested in any of the CHAMP1 families that might have some signs of any kind of blood abnormalities, anemia or leukemia or low white blood cell count or immune defects. I'd be particularly interested in knowing about that. Talk to me after or at lunchtime. And then the patients also have a very characteristic cellular appearance. Their cells are very sensitive to radiation, like CHAMP1 deficient cells, very sensitive to a drug called mitomycin C, and they develop these special kind of chromosome breaks. And that's how I diagnosed the disease. So my laboratory in Boston is the North American center for diagnosing this rare disease, Fanconi anemia. So this is a really important slide, because this goes to show exactly what you're doing right now. So for the last 30 years, every year, except the last 2 pandemic years, every year for the last 30 years, I've attended a camp in the summer where we collect families from around the country, now around the world, several families from Brazil actually, to a camp in Maine, and we bring the children, the families there, we teach them about the disease, we learn a lot about the disease. Some of the most important discoveries I've made in Fanconi anemia came from talking to families at this camp every summer. We just learned things we just didn't know as scientists. And then, of course, we collected blood samples and skin samples. Sometimes we did it right on the spot. We would actually, one of the parents was a podiatrist, and she helped me collect skin samples from the children and the family members. Now, of course, you can do it by mail and do it with some of the organizations that you heard about this morning. But back then it was tougher. We used to bring a kit with us to the camp and collect the blood samples right on the spot. So incredibly important what you're doing right now. And over the years, what we found is there's now 23 genes for Fanconi anemia, okay? I would argue for CHAMP, there's 4 genes that are of interest right now: POGZ, HP1 alpha, CHAMP1, and Rev7. There are now 23 Fanconi anemia genes, and really my career and what I'm probably best known for is assembling these genes, assembling these proteins into a pathway called the Fanconi anemia pathway. And here's the pathway here in very broad strokes, that these 23 proteins cooperate in a common pathway, just like POGZ and CHAMP1 do. And in this case, those 23 Fanconi anemia proteins cooperate in a systemic pathway which is required for DNA repair. And if there's a defect in any one of these 23 genes, the cells are sensitive to DNA damaging agents like radiation and like mitomycin C. So it's a pathway, and knocking out Rev7, which is one of the critical proteins in this pathway, it's a Fanconi anemia patient that has mutations in Rev7, results in this defect in the pathway, okay, and cellular sensitivity to cross-linking drugs. Now, I would just like to argue today that CHAMP1 also has a pathway, just like Fanconi anemia pathway on the right, a series of 23 proteins working together. On the left, CHAMP1 works, the protein works with POG, it works with HP1 alpha, it works with Rev7, it works with another protein called TRP13, and this pathway seems to be important for chromosome alignment. But as you just heard elegantly from Kozo, and I didn't know this work because it's unpublished, is that this pathway on the left also participates in gene transcription, which is really interesting, and thank you for sharing that, Kozo. So, let me tell you about Rev7, because it's such an interesting protein to me. It's a small protein. We like small proteins. They're easier to work with. It's 20 kilodaltons in size. It's an adapter protein. It actually exists in 2 forms. So the Rev7 protein is closed and open, closed and open. And when it's closed, it binds to other proteins. It has a seatbelt. So when Rev7 closes, it binds to Rev3. That's a protein involved in translesion repair. It sometimes binds to a protein called SHIELD3 that's involved in another DNA repair pathway. And the exciting finding is number 6, the Rev7, when it closes, it latches on to CHAMP1. And this is a regulator of a certain kind of DNA repair called homologous recombination repair. And this, as I showed here, the Rev7 protein also binds to this complex here, which is called the Shieldin complex, which is involved in non-homologous end joining, and so another repair pathway. Now, let's see if that went back forward. Yeah, so we thought, wow, that's so interesting that Rev7 is so integrally involved with different DNA repair pathways. We asked the question, if we pull down Rev7 in a cell, does it bind to other DNA repair proteins? Well, when we did that, what we found was that it binds to a protein called TRP13, which shown here, what TRP13 does is it's a protein that, it's a machine that opens and closes the Rev7 protein. And when it opens Rev7, it releases the CHAMP1 protein. Very interesting. So a summary of all this then is that common number 2 here, TRP13 opens the Rev7 protein, releases SHIELD3, but it also releases the CHAMP protein. And this switches cells around from one DNA repair pathway to another. Okay. All right, so we said to ourselves, okay, this was Feng Li, a very talented postdoc in my laboratory. He asked the question, if Rev7 is so critical to these DNA repair pathways, maybe if we pull it down, we'll find that it binds to another protein, some other protein involved in DNA repair. And look here with this arrow. Turns out one of the most prominent proteins that Rev7 binds to is the CHAMP1 protein. And another protein it binds to, the POGZ protein. So these are your friends. These are the proteins that your society is so interested in. So we found from this experiment, when we pulled down Rev7, it binds to CHAMP1 and it binds to POGZ. So we're connected. All of us are connected now through this protein-protein-protein interaction, okay? So this is Fang Li, who's the postdoc in my lab, who's done a lot of this work. He's very fortunate. He came to my lab right before the pandemic, and he found it wise to just live in the laboratory during the pandemic. He has a cot there and did a tremendous amount of work with us. And this is the paper that he published, which really this paper, he's the first author, Fang Li. He showed that the CHAMP1 protein binds to Rev7, promotes DNA repair, and I'm really pleased to say that this paper came out right at the same time that Kozo's paper came out, and we said the same thing. And that's a really important thing in science when two independent laboratories from around the world come up with exactly the same conclusion. That kind of validation is really central to everything we do in science, is to have independent validation. So, yeah, so think of this now. So Rev7, this weird little protein, 20 kilodaltons, a little guy, a very abundant protein, it binds to different cargo. Sometimes it seatbelts around a protein called Rev3. That causes a certain kind of DNA repair. Or in the middle, Rev7 sometimes binds to a protein called Shield3, and that causes a different kind of DNA repair. And now on the far right, Rev7, its seatbelt grabs onto the CHAMP1 protein, and this regulates a very special kind of DNA repair called homologous recombination. And we prove that. We prove this because we looked at the CHAMP1 protein now, and we found that the CHAMP1 syndrome has some phenotypic, some characteristics which are similar to Fanconi anemia. There's common cellular and clinical features. There's the protein, 812 amino acids in size, named after Kozo's group, chromosome alignment maintaining phosphoprotein. CHAMP1's required for a certain kind of chromosome alignment that he just referred to. The CHAMP1 mutation is as you know, only one allele, one gene is mutated, the other gene is wild type, okay? And CHAMP1 binds to this Rev7 protein, and the crystal structure was closed. When I entered this field, really the only papers that were published in this field were of special meaning to me, really came from Kozo's laboratory. So in fact, it's almost, to a scientist, it's almost a special treat to come into a laboratory when there's only a few really good papers published, because it gives us the ideas of how we want to proceed. Sometimes it's more complicated when you enter a field and there's thousands of publications, and a lot of those publications are wrong. In the case of the CHAMP1 field, there are only a few publications, but boy, were they right. Kozo got it right. And one of the things that I found was that there are very rare patients with CHAMP1 syndrome that do have some blood abnormalities. And I won't say who the families are, but I've been talking around, and there's some cases of white blood cell count low, there's some cases of leukemia, there's some cases, and this intrigues me because this makes the clinical situation, the similarity between CHAMP1 syndrome and Fanconi anemia even stronger, okay? Now, I'm just going to show you a few scientific points. One is that when we knock down the CHAMP1 protein, use a tool called CRISPR, which I'm sure you've all heard of, that you can genetically knock down a gene in a normal cell. When we knock down the CHAMP1 gene, we found indeed that these cells have a DNA repair defect, a special kind of DNA repair defect called a homologous recombination defect, which again, my laboratory happens to be an expert in. Now, I can skip that. Now, one of the other things we found, and this is really more for the scientists in the field, but I think it's worth pointing out that in the audience, CHAMP1 promotes homologous recombination by regulating this specific complex. So what I show here is, on the right-hand side, this bar graph that normal cells have elevated level of homologous recombination based on the level of Rad51 foci, which is a measurement. It's a microscopic measurement of DNA repair. If you knock down CHAMP1, okay, with a so-called siRNA, now those cells have very low levels, the bars are very low, of Rad51 foci. They have very little homologous recombination repair. If you knock down a gene called Rev7, shown here on the right of this bar graph, you get a very high level of homologous recombination, okay? But even if you knock down both Rev7 and CHAMP1, you still find a high level of homologous recombination. And that told us that, in fact, CHAMP1 is doing a very specific function here. The CHAMP1 protein is somehow suppressing this Rev7 complex, and that Rev7 complex normally suppresses homologous recombination. So what CHAMP1 is doing here, its function is by suppressing the Shieldin complex, it's actually promoting homologous recombination. So we have a mechanism here. Again, a mechanism that was largely validated from Kozo's work. Okay. All right. Now this is a so-called immunoprecipitation, again, primarily for the scientists in the room, but I can describe it, I think, simply. You can see on the left, this is a so-called autoradiogram, that when we knock out the CHAMP1 protein, see with the arrow there, you see an increase of binding of another cargo protein, SHIELD3, to Rev7. So knocking out CHAMP1 results in a switch now to a different DNA repair pathway, the binding of Rev7 to so-called SHIELD3. And this kind of protein-protein interaction is a very commonly used tool in my laboratory. Okay, there we go. All right, so this was probably the most interesting point that I just gleaned from Kozo's talk, which is I've been thinking about the CHAMP1 complex, shown here at the top, CHAMP1 bound to POGZ, bound to HP1 alpha, as a complex that binds to heterochromatin, certain regions of a chromosome, like centromeres in the middle, or the ends of the chromosome called telomeres. But what we just learned from Kozo is this complex also tends to bind specifically to sites that activate certain genes, genes involved in cell division and genes involved in brain development. So this is fascinating. This complex of proteins, including the CHAMP1 protein, binds to these discrete regions, but also plays a very new role in transcriptional activation, which I'm sure we'll be hearing more about from Kozo's laboratory. Okay. Now, all right, so this is interesting. There have been a lot of points that already came up this morning from certain families. We heard from a question about it just now, about missense mutations. Most of the CHAMP1 mutations, as you all know, are nonsense mutations resulting in truncated proteins, right? But in fact, there are a few families that have point mutations, missense mutations in the full-length CHAMP1 protein. And it's kind of difficult to know whether or not those point mutations are really pathologic mutations or are they so-called variants of unknown significance. Well, here's a test. For instance, in this experiment, what we did is we took the full-length CHAMP1 protein, okay, and we made a single point mutation, or right in this Rev7 binding motif, okay? And this amino acid change, this single amino acid change, now blocked the ability of the CHAMP1 protein to bind to Rev7. And that's important because that says to us this point mutation, this missense mutation, really has functional significance. So we'd be very interested in testing missense mutations from a couple of the families in this room to see if those missense mutations also prevent the binding interaction to the Rev7 protein. Okay, because these are functionally significant mutations which prevent homologous recombination repair from happening. Okay, now I'm really not going to say much about this slide at all other than to say that the CHAMP1 protein has new vitality and new interest in the cancer research field. Okay, obviously, I mean, I'm not a specialist at all in intellectual disability or in brain development. That's not my shtick, that's not my thing. But I am an expert in cancer, and I'm an expert in blood cancers in particular. And now that we know that CHAMP1 protein is somehow connected with Rev7 and DNA repair pathways, suddenly your syndrome has broad interest to the cancer research community. And that has important funding implications, that has important broad relevance, that'll bring new scientists into the field. This is good. This is good for this community. You will have more scientists showing up here. You'll have more money. I can't say it's going to flow into your society, but I think it'll come a little bit faster than that you've been able to raise so far. Okay. All right, so conclusion of this then is Rev7 CHAMP1 is a positive regulator of this neat DNA repair pathway called homologous recombination repair. The CHAMP1 binds to Rev7 and prevents its assembly with other DNA repair proteins. The CHAMP1 overexpression is common in tumors, cancers, and this is where the CHAMP1 protein, when deficient, can cause some mental disability, but when overexpressed can actually cause cancer. And that's where the cancer community will find interest in this. And yeah, so we'll skip on then. All right, so this kind of like a similar conclusion of what I've said is that this Rev7 protein, which is near and dear to my heart at the top here, it binds to a few different complexes. On the left, it binds to CHAMP1. On the right, it binds to the Shieldin complex. And these two complexes are competing with each other for Rev7 binding. And they're regulating DNA repair accordingly. So CHAMP1 is a novel DNA repair protein that regulates homologous recombination. Damage induction can actually promote the interaction of CHAMP1 and POGZ, very consistent with what we just heard about from Kozo, that knocking out CHAMP1 results in radiation sensitivity of cells. And then lastly, CHAMP1 POGZ complex sequesters Rev7 and prevents it from forming other DNA repair complexes. I'll skip that. So as we've heard, I'll just say it again, CHAMP1 protein interacts with POGZ and another protein called HP1 alpha. And in this forms a so-called heterochromatin complex. And this complex is actually well known. It was actually discovered 12 years ago by a paper published in the journal Cell that CHAMP1 binds to POGZ and binds to HP1. Of course, we didn't know at the time what CHAMP1 was actually doing. Now we know that knocking out any of these proteins in this pathway, and maybe this is the most important thing I'll say to you, I think CHAMP1 is not an isolated protein, it's part of a pathway of proteins. And knocking out any one of the proteins in this pathway is going to result in a similar syndrome, a syndrome of mental disability, a syndrome of DNA repair defection. And I think that knowing it's a pathway actually provides an opportunity for ameliorating this disease and coming in with drugs or gene therapy that could counteract this defect. And these proteins function in the same pathway. Now we're going to call it the CHAMP1 pathway. Okay, so this is my last slide. What I've told you is CHAMP1 syndrome is a DNA repair syndrome, okay? It's a disorder of DNA repair. Rev7 regulates these three distinct pathways that I've told you about: TLS repair, non-homologous end joining, and now homologous recombination. That Rev7 binds to CHAMP1 and regulates HR repair. That humans with CHAMP1 syndrome, individuals in this room that have mutation in one of their two CHAMP1 genes, have what I would consider a mild DNA repair defect. It's mild when it comes to certain blood abnormalities, but it's quite severe, obviously, when it comes to cognitive issues and speech issues. So the implications of this are very important clinically. It means that CHAMP1 patients may have symptoms or signs of DNA repair disorders. Mild blood abnormalities, that's why I reach out to you. Let me know if anyone's had mild, if their children or young adults have had these defects. Sensitivity to sunlight, sensitivity to certain dietary things that cause DNA damage, sensitivity to certain drugs. If any of the CHAMP1 patients are on seizure medicines and can't tolerate those seizure medicines, it could be because the seizure medicines are causing DNA damage that cannot be repaired because of the CHAMP deficiency. Developmental abnormalities are common. And the intellectual disability of CHAMP1 syndrome is really distinct from other DNA repair disorders. Fanconi anemia and some of the related disorders, we do not see as much in the way, we see microcephaly, small heads, but we do not see intellectual disability the way we see in the CHAMP1 syndrome. And that's interesting, and that's a challenge to us. All right, so here are the people in my laboratory. This is Jeff Shapiro, who's a very, very close colleague of mine. We run a Center for DNA Damage and Repair at Boston Children's Hospital and Dana-Farber. So we're interested in diseases. Now we're very interested in CHAMP1. And these are the individuals in my laboratory right now, especially Feng Li, who fortunately spent the pandemic with me. And I think I'll stop and answer questions. Thank you.
I have quite a few questions. I'm going to save them. It's me. I'm going to save them maybe till after lunch, where we have a few extra minutes.
Yep.
There's also a couple questions someone had reached out to me to ask you, but I'll just save it for two because we have a virtual speaker next. I know they've been waiting, so we'll take two or three questions now, and then we could save the rest for after lunch.
Or over lunch. Yeah.
Thanks. This is really interesting work everybody's doing. Just a very simplistic question. So if CHAMP1 suppression of Rev7 complex is what's necessary, would it be possible to knock down Rev1 in CHAMP1 patients as a potential clinical treatment?
Yeah. Yes.
Nice.
Yes. I mean, short answer is yes. That's why we'd like to know the whole pathway. We'd like to know the whole pathway because possibly modifying the bottom part of the pathway might help us with the loss of CHAMP1.
Do you already have a small molecule that knocks down Rev1?
No. No, we don't. We're working on it.
Cool.
Just a very, very quick one, Alan. Really elegant work. I mean, obviously, with DNA repair mechanism being disrupted, the concern is that this one of the mechanisms of preventing cancer is that a body has a robust repair mechanism. Is this yet another concern now that we have to face and maybe screen and assess?
It is a concern. And I think that this repair pathway, the CHAMP1 repair pathway, is more vital to neural development, transcription in brain development, less important to regulating DNA repair in other tissues. So I'm not expecting, certainly not hoping, to see any cancers at all in these families, but boy, we're going to be looking for it. Because, as you said, loss of any DNA repair pathway, a Fanconi pathway or the CHAMP1 pathway, will result in genomic instability and cancer predisposition. Sometimes just anemia, and that actually becomes the first sign that a cancer may be developing. And so I think that, thank you, we're attentive to that. That's why the registry will be of considerable importance. And if a child with CHAMP1 syndrome were to develop an early cancer, or a young adult were to develop an early cancer, there are ways to be surveillant about that and prevent that from happening. And because cancer surveillance and cancer prevention are huge fields now in the cancer research field. So we would want to know. We would certainly want to know.
Yes, quickly. Our child seems to particularly meet a lot of the things you find interesting, right? A lot of the key points up there you said. How do we go about, I guess, providing you additional data, like more ammo for your data set? Because he did seem like pretty much all of the keywords you were digging into, that's our child.
Yeah. Through the registry and through lunchtime conversation, but through these meetings. I told you every year I went to this camp. My biggest discoveries in my laboratory came from conversations with parents of children with a DNA repair disorder. So thank you for that. Yeah. Can you yell it? Yell it.
Do you see any kind of do you see any kind of issues with the immune system, like T cells, B cells, and so on?
Yeah.
Talking about cancer, right? So, and like eventually the potential to do like cell therapy, like T cell, CAR T cells, and so on.
Right. We'd be very interested in knowing whether or not any of the children or young adults with CHAMP syndrome have any kind of immune abnormalities, recurrent infections, low white blood cell count. I don't think this is going to be common. Frankly, I think we know about it already, just from the table we heard this morning. But I think that there are occasional patients that might have it, and I think this would be really important to know. One of the most common, simple features of a DNA repair disorder to monitor is the blood cell count. It's a simple way to measure that. Yeah.
Thank you.